Okay. How is, what is tissue fluid? We begin from there. Remember that tissue fluid is that fluid surrounding cells, or a fluid we find within the tissues. A tissue is a group of cells that usually perform a similar function. Look at that visual. That image there is giving us a summary of what or where exactly we find the tissue fluid, and it is going to help us in the formation of tissue fluid. In our transport, we saw that blood from the heart, or blood is pumped by the heart through blood vessels called the arteries to the tissues. Then the tissues are drained by veins. But we saw that when blood moves through arteries towards the tissues, the arteries divide into narrower blood vessels known as arterioles. Arterioles further branch into narrower blood vessels known as capillaries. Capillaries later join and form what we call the venules. The venules then form the veins that return blood to the heart. Meaning, from the heart, blood flows through arteries to narrower vessels called arterioles to capillaries, which capillaries penetrate through the tissues. Then capillaries give rise to the venules give rise to the veins. This therefore means that which blood vessels do we find in the tissues, penetrating the tissues? These are the capillaries. And as such, as blood flows from the arterial, capillary, venule, vein, heart, and it continues. When blood reaches the capillary, remember, capillary walls are semi-permeable. They allow some molecules that are small to pass through. But we also noted at a particular point that capillaries are selectively permeable. Selectively permeable to mean that they allow particular molecules to pass through at one moment, but they may restrict them to pass through at another moment. Therefore, they are selectively permeable. Remember from transport, where we saw that capillary walls are very thin, almost one cell thick, in order to offer a short distance over which materials will diffuse across them. Equally, even here, when blood is flowing within the capillary, just like we are seeing there, materials that are made of very small molecules are able to pass across the walls of the capillaries from blood into the tissues where the cells are, into the tissues, and they surround the cells. These materials with small molecules include oxygen, glucose, amino acids, fatty acids. We talk of hormones, ions, antibodies, and some white blood cells. But those that cannot pass through the walls of the capillaries include the red blood cells, because they are large. Include some large plasma proteins. There are some plasma proteins that are large, big. Therefore, they cannot pass through the walls of the capillaries. Meaning that materials move from blood in the capillary across the capillary walls into the tissues, forming a fluid called tissue fluid. But these materials have small molecules. We've given examples: glucose, oxygen, amino acids, fatty acids, hormones, ions, antibodies, some white blood cells. But those that are not allowed to pass through are those that are large. including red blood cells and other plasma proteins. What does that mean? That at that point we have some filtration, and that process is what you call ultrafiltration. Ultrafiltration, allowing some molecules to pass through while others do not pass through. By the way, molecules that don't pass through blood cells, but we've said that some white blood cells are able to pass through. Why? Because white blood cells are amoeboid. Some white blood cells are amoeboid. Amoeboid to mean that they keep changing their shape. Now when they change their shape, being amoeboid, they are able to squeeze through the tiny pores of the capillary walls, therefore getting into the tissues, getting into that part where they surround the cells. That is why tissue fluid is also called the intercellular fluid. Why? Because the fluid does not enter the cells, but it is found in between the cells. That's intercellular. Also called extracellular, extra outside, extracellular fluid. Extracellular, meaning this fluid is found outside the cells. But remember, because a group of cells is what we are calling a tissue, that is why we are calling it tissue fluid or extracellular fluid or intracellular fluid. And we've seen that its process of formation involves ultrafiltration. Ultrafiltration. We now move on. Tissue fluid formation involves basically two major forces. Two major forces that play a big role. One of them, there is a pressure which we call hydrostatic pressure. Hydrostatic pressure. And the other is called osmotic pressure. Attention here, much attention here. Hydrostatic, hydro, fluid. Therefore, hydrostatic pressure, is that force that arises within the fluid called blood. But that force in blood is as a result of the pumping action of the heart. Meaning hydrostatic pressure, that force due to the fluid, because blood is a fluid, that pressure that is developed within blood as a result of the pumping action of the heart is what we are calling hydrostatic pressure. This means that hydrostatic pressure is very important in forcing these molecules that are small from blood across capillary walls into the tissue. But there are molecules that remain behind, the large ones, protein molecules and the red blood cells. Now those protein molecules which remain behind in blood, the ones that are not filtered, the ones that don't move out of the capillary, those protein molecules now bring about a force, a pressure which we are calling osmotic pressure. And it is a pressure that tends to pull back water molecules from the tissue fluid back to the blood within the capillaries. Therefore, in formation of the intercellular fluid or tissue fluid, we've said two forces are important. One, hydrostatic pressure, and two, osmotic pressure. Remember, we said that tissue fluid is formed by ultrafiltration, where as blood passes through the capillaries. As the blood passes through the capillaries, that tissue fluid formation is controlled by two major processes, hydrostatic pressure and osmotic pressure. Hydrostatic pressure due to the force from the heart and the capillaries, while osmotic pressure is due to the proteins, the plasma proteins, which remained in blood, within the blood capillaries, that tend to pull back water into the capillaries. Therefore, Moving forward, we are going to see that the capillary has an end near to the arterial and it has another end near to the venule. The end near to the arterial is what we call the arterial end, and the one near to the venule is what we call the venule end. Meaning there is even that part in the middle, in between the arterial end and the venous end or the venule end. Now, after noting that we have those two pressures, how do they interact? This now brings us to the actual formation of tissue fluid.